Hello, and welcome to our second section on the human eye. This will be the human eye, part 2, and today we're going to be talking about the human eye and phototransduction. Subsequently, we'll move on and talk about a particular kind of cell in the human eye, ganglion cells. So we'll have our introduction to ganglion cells, but first we'll start with phototransduction. So embryologically, that is, when you are an embryo, we can say that the retina was originating from the same tissue that spawned all of your neurons. And this is an interesting fact about the eye, and that is that unlike many other kinds of senses, the eye is actually considered part of the brain. And it's considered part of the brain for embryological reasons. That is, unlike your ears, your eyes actually were part of the same tissue that spawned all subsequent neurons. So for this reason, a lot of neuroscientists will consider the eye to be actually part of the brain. Like other brain cells, cells in the retina cannot be repaired. So some of you may be aware of an important disease known as macular degeneration. And this is an important disease because sight is so important to our everyday lives, but also because once we begin to lose photoreceptors, cells inside of our eyes, it's very difficult to recover those. Generally, they're gone for good. There have been some new therapies in recent years, but we're still not all the way there. Right? Most importantly, the retina is located, or is a location of phototransduction. So an interesting concept and an important concept in sensation and perception is that we have to, in some way, be able to convert environmental energy into neurons firing. And the neurons firing presumably are going to roll up into some kind of a phenomenological experience that we would have. And we talked previously about some issues in the philosophy of science and the philosophy of mind, where we try to understand how it's the case that we can get from neurons firing to the experience of red, or the experience of a saxophone. Before we get to that problem of having neurons firing and converting that into some kind of phenomenological experience, we have an intermediate step, or an earlier step, I should say, of converting environmental energy into axons firing. And this is a transductive process. We're going to transform energy out here in the environment into some kind of neurological code. So this is transduction, and when it's happening in the world of light, we call it phototransduction, so that you can see in that word phototransduction the root of photo, referring to photon, and transduction, to transform energy from one state to another. Okay. So we can say then that photoreceptors are facing the wrong way, and this is an interesting factoid about our eyes. In fact, we're going to understand two ways in which the construction of our eyes is a little bit counterintuitive. Two ways in which we might say the eye is perhaps backward relative to the way that you might have thought about it. So let's take a look at this diagram. And in this diagram, we'll just orient ourselves. Here I have the word anterior, and here we have posterior. So I'll point along with you. Inside of our heads, we can say that the front side is, or the front end, is going to be our anterior end, or our anterior side, and toward the back we have posterior. So we have anterior to posterior, and we could talk about that even within the eye. The front portion of the eye would be a relatively anterior portion, and the back portion would be a relatively posterior portion. So in this diagram, it's just a little bit confusing. Light will be entering from the top. Okay? And the photoreceptors, which are going to be doing the transduction for us, they're going to be, if you will, catching the photons. Very interestingly, they're actually pointing away from the incoming light. Now that's counterintuitive. You might have imagined it would have been perhaps more effective to have the photoreceptors pointing right at the light as opposed to away from the light. But that's not the way our eye is constructed. It's constructed very differently. Okay? So here's the anterior portion, here's the posterior portion. And light is coming in. Interestingly also, light has to pass through all of this neural machinery before it reaches our photoreceptors. So we have some neurons up here that we're going to come back to. These are called ganglion cells. The ganglion cells themselves have to be of a very high optical quality. And what we mean by that is light has to be able to pass through them. So these cells are going to play a very important role in just a little while when we move further into our story. For right now, what we need to know about them is light is actually passing right through them. They are, relatively speaking, transparent. And then the photoreceptors are going to catch those photons, even though the photoreceptors are pointing away from the light. What actually happens is the light is going to bounce off of this back end, this most posterior portion of the retina, and those bouncing photons will become available to the photoreceptors for transduction. Okay? So this is the first instance of something being backwards inside of our eye. Photoreceptors are facing away from the incoming light. By the way, that isn't true for all animals. It happens to be true in the human eye, but not for all animals. There are two categories of photoreceptor, and this you might remember from Intro to Psych. We talk about rods and we talk about cones. Okay, these are cells, and they're going to contain different kinds of opsins, which are proteins. But let's first just understand these two broad categories that we have of rods and cones. So rods are far more numerous. They outnumber by approximately 100 million to 5 million in each eye. So we have way more, or far more, rods than we have cones, and there's the ratio, about 100 to 5. Okay? So you might think of cones as being relatively precious because they're relatively rare. The most central part of the retina contains only cones, so the rods and cones are not equally distributed across the expanse of your retina. We have a high concentration of cones in the center of the retina, which we call the fovea. Okay? And then as you move out from there, you will have fewer cones and more rods. Okay? In the periphery, and the far periphery especially, when you moved way out into the outer reaches of your retina, we have only rods. Lumping rods and cones together, photoreceptor density overall decreases with eccentricity, and what we mean by that is moving further and further away from the center of the eye. So vision is less sharp in the periphery. That is to say that we have fewer photoreceptors per unit space as you move away from the center of the eye, and as you move toward the center of the eye, it's kind of like moving toward the center of a downtown city. The population tends to grow, and as you move out from the center of the city, things become a little bit more sparse in their density. Okay, so for vertebrate eyes, you and I are vertebrates, which is to say that we have backbones. There's a very interesting dichotomy in vision science between vertebrate vision and invertebrate vision. And for us vertebrates, light increments lead to hyperpolarization, and light decrements lead to depolarization. This is exactly opposite from the way that it is in invertebrates. So we have very different patterns. I'd like to just unpack that a little bit for us. Specifically, I want to unpack what is meant by light increments. Through the years, I found that students sometimes are confused by that. So in the classroom, one of the things I will do is I will walk over to the light switch and just throw the lights on and throw them off, throw them on and throw them off. When I'm doing that, I'm creating light increments when the light is turned on and light decrements when the light is turned off. What this phrase is telling us is that when we turn the lights on, we actually have more light and less neurotransmitter being released. The transmitter of interest is a transmitter in our eyes called glutamate. So when we turn the lights up, because you and I are vertebrates, we actually get a decrease in glutamate release. Okay, so lights going up means less glutamate, and then when I turn the lights down, we actually get an increase in glutamate release. So this again is backwards. So maybe the second idea that we've had about something being backwards inside of our eyes. Okay? Now in the invertebrate world, it's a little bit more intuitive. We turn the lights up and we get an increase in glutamate release. We turn the lights down and we get a decrease in glutamate release. So the invertebrates seem to have it in a way that's perhaps more intuitive than what you and I have. But it is interesting that these two different mechanisms have evolved. And this harkens back just a little bit to what David Marr was talking about in his 3 levels of analysis for vision. This very bottom analysis would be what we would call the implementation level. And the way that we as vertebrates respond to light is very different from that of invertebrates, and the difference is in the way of implementation. Where we're having hyperpolarizations, they're having depolarizations, and so forth. Okay. Let's talk a little bit about these opsins. Opsins are proteins, and there's an opsin inside of rods, and there are opsins inside of cones as well. The opsin inside of rods, we call that rod opsin. That's easy to remember. It sounds like rod opsin. It responds best to a particular range of wavelengths. And in our previous section, we talked about different wavelengths of light. You might remember that the human animal is quite good at responding to wavelengths of light that range from approximately 400 nanometers on the one side to 700 nanometers on the other side. So that range is pretty accessible to our eye. And these particular photopigments, these opsins, are going to respond best to about 590 nanometers. Okay, that's their peak sensitivity. They can pick up other wavelengths as well, but they're particularly well tuned to that wavelength. When we talk about the cones, we actually have three different kinds of cones, three different kinds of cone opsins as opposed to rhodopsins. Three different varieties, at least most of us with what we might call typical human vision or trichromatic vision will have these three cone types, okay, cone opsins. And I would like you to memorize these numbers, please. They're approximations, but just to get the ball rolling, we have one type of cone opsin that responds maximally to light at 440 nanometers. A different kind of cone opsin responds maximally to light that's coming in at 530 nanometers, and then the last one is responding best to 560 nanometers in their wavelength. Okay? Now, a difficult concept for some students is this concept called the principle of univariance. So we'll read it once, and then we'll see if we can understand it. At the point of phototransduction, that is to say, when a photoreceptor actually catches one of these photons, at whatever wavelength it might be one of these wavelengths exactly, more likely it's going to be off of those wavelengths by a little bit, but when we catch that photon, the point of phototransduction, at that moment, all wavelength information is lost. So a very interesting idea about this phototransductive process is that the photoreceptors are initially in one formation, and they change their formation. And this is called a cis-trans isomerization. So typically, the photoreceptors will start out in a cis form. And when a photon is caught, this actually transforms or changes the shape of that photoreceptor, or actually that opsin. And so we're going to go from this shape to that shape, and you might do this with me. We have a cis-trans changing shape, which we call an isomerization. So we go cis-trans isomerization, and we can do that now with different kinds of rhodopsins and different kinds of cone opsins. And once that cis-trans isomerization occurs, we're going to get some change in the level of glutamate that will now be an important factor in downstream processing. But this is what gets the ball rolling. We catch the photon, we have the cis-trans isomerization, and once that occurs, though, we've actually lost all information about the wavelength of light. Okay? We lose all that information. It's not like we get half of a cis-trans isomerization if we get one wavelength of light, and we get a full cis-trans isomerization if we get a different wavelength of light. We either get the cis-trans isomerization or we don't. Okay? And we lose all wavelength information. This is called the principle of univariance. We have a single response. Univariant single response. Here's one of the ways of thinking about this. It's an analogy that has been helpful to some of my students through the years. You and I can be looking out your front door, and we might see that there is a cat that's walking by. At another moment, we might see that there's a rabbit that's walking by. And you and I can describe the cat as a cat. We can describe the rabbit as a rabbit because you and I can make two different responses there, cat or rabbit. Okay, we can do that. But suppose it were your dog. Your dog might be inclined to bark at something that's hopping or moving in front of the front door, and if the dog sees a cat, the dog will bark. If the dog sees a rabbit, the dog will bark. Two very different stimuli out there, yet we have a univariate response. The dog is basically barking. Okay? And that's all the dog can do. It can only make that single response. Back to our photoreceptors, we can get a cis-trans isomerization in response to a photon, if a photon is present. But we're not able to distinguish with the cis-trans isomerization whether that was a cat or a rabbit, if you will, which is to say we don't distinguish between a 440 or a 530 or any particular wavelength of light that's most likely going to land between 400 and 700 nanometers. So that's the principle of univariance, and we'll come back to that. What I'd like to do now is offer you something akin to David Letterman's top 10 list. And in his top 10 list, he has some kind of a theme, and he works his way on down. We're going to have a top 3 list, and these will be the top 3 reasons why it would be quote-unquote vulgar to refer to human cones as red, green, and blue. So in Intro to Psychology, you were probably taught that we had 3 cone types, and we called them red, green, and blue. And as is true in many educational settings. That's a perfectly reasonable way to get the conversation going. Sir Isaac Newton, though, once used the phrase vulgar not to necessarily mean offensive, but he meant commonplace, perhaps overly simplistic. And it is really commonplace and overly simplistic to refer to our three cone types as red, green, and blue. And we're going to understand three reasons why that's the case. The number 3 reason is this. Newton said so. That is to say that you might remember from the previous lecture, Newton reminded us that the rays are not colored. So if we say that we have a red, green, and blue cone type, then what we would be saying is that somehow the rays of light are red or green or blue, and that's really a stimulus error. The rays of light differ from each other in their wavelength, they differ in their energy levels, but they really are not colored. So when we refer to a cone as catching a red wavelength of light, that's really a misnomer. Okay? So our first reason then is Sir Isaac Newton said so. Okay. And you might remember also that Billy Wooten reminded us that the red's in the head. The red is not out here in the real world. Okay, let's go on to the second reason why it would be vulgar or commonplace or overly simplistic to understand or to use the terminology of red, green, and blue photoreceptors. Right? The peaks don't match perception. Right? And what we mean by this is the peak wavelengths don't match our perceptual experiences. Let's see if we can understand how to unpack this. I asked you a moment ago to begin memorizing these 3 peak sensitivities that characterize our 3 cone type responses. They were 440, 530, and 560 nanometers. 440, 530, 560. If we look at 440, and if I were to show you light that was a laser consisting only of 440 nanometers, it would look violet but not blue. And yet we call this the blue photoreceptor. And that's a bit of a misnomer for that reason. This peak sensitivity doesn't match our perceptual experience. Okay, we can go to the 530 nanometer light, and it looks green. And that's actually a pretty good match. We got lucky on that one. So the photoreceptor that we call green actually does line up with the nanometer designation that we have here. If we go to 560, though, if you and I were to look at a 560 nanometer light, it would actually appear yellow, not red, and yet we call that our red cone. So we don't have a real good match, at least in 2 of these 3 cases, between the wavelength of light that's actually being absorbed and our perceptual experience of that. So this is yet another reason why it's a bit of a misnomer to call the cone types red, green, and blue. Okay? And then here's the last reason. We've introduced this once before, but because it's a complicated idea for many students, I wanted to introduce it here again. And that is the principle of univariance, which we said a moment ago, that all wavelength information is lost at the point of phototransduction. So the idea would be this. We have a photoreceptor that might be maximally sensitive to something like 440 nanometers of light. But when a cis-trans isomerization occurs there, it could occur really in response to any kind of photoreceptor, any kind of wavelength of light. Okay? It could be 440 nanometers, but it could also be 400 nanometers. The cone type that we have here can catch photons of any wavelength. It just happens to tend to be most sensitive to this wavelength of light. But if we do get a photon at, say, 700 nanometers that's caught by this particular kind of photoreceptor, we're still going to get the cis-trans isomerization, and we're still going to have the change in glutamate levels that we would normally get. So we've lost some precision in what it is that's been caught. We have this principle of univariance. Any cone can absorb any wavelength. The differences that we saw across the cones were just their statistical tendencies. Okay, so no vulgarity, please. We'll try to avoid speaking of the red, green, and blue cones. Okay, let's go on to a learning check. So we've talked about a lot of information in part 1 and now in this introductory portion of part 2. And what we can say is that we want you to do the following. We'd like you to be able to describe the 3 types of refractive errors that we had talked about in part 1 and the lenses that are used to correct them. This is a very practical exercise. Throughout your lives, you'll be running into people who have very different kinds of refractive error. In your own development, your refractive error will change as you age. It actually changes almost from year to year. Okay? So it would be helpful for you to know these different kinds of refractive errors and the kinds of lenses that we use to correct those. It would also help if you could be able to identify the 3 reasons why it's inaccurate or quote-unquote vulgar, commonplace, to refer to our 3 cone types as red, green, and blue. You might review what we just talked about. And then you might go back to the end of our last video where we talked about the blind spot. And we'd like you to be able to find your blind spot on a regular basis. Okay? So I'll give you a moment to stop the video and see how you can make some progress on these kinds of issues. Okay, let's go on to our next topic, which is an introduction to a different kind of cell type, the retinal ganglion cells. In our previous section, we talked a little bit about photoreceptors, and these are the cells that actually perform sensory transduction, and specifically phototransduction for us. These are the ones that catch the photons and start up the sequence of events that will convert physical energy out here in the distance to some kind of action potential inside of our nervous system. So those were photoreceptors. We talked about the cis-trans isomerization. Let's go a little further in the physiological response to light by talking about retinal ganglion cells. So we're still inside of the retina, which is where the photoreceptors are. Let's see what we can do here. Here's a big picture of the eye. And note that the retina is at the posterior pole. So light is coming in this particular diagram from this side. And back here on the fovea and all the way back on the retina, we have the photoreceptors. And then we can zoom in and take a look at what's going on even inside of the retina. Okay? So the retina is going to consist of 3 different layers. We've talked about the photoreceptors. We mentioned that we had rods and we had cones. The cones themselves had 3 different varieties, at least for our species. For other species, there may be more or fewer varieties of cone. Then after the photoreceptors catch the light, there's going to be some activity in a middle layer of cells. And there are 3 different cell varieties here, the so-called horizontal, bipolar, and apical. And they're going to operate in that sequence before we get some activity in the next layer of cells called the ganglion cells, which is what we're going to be focusing on predominantly in this section. So let's see how all of these are arranged. Okay? We're back to a diagram that we had seen just a little bit earlier in this video. We talked about how light is coming in over here. I've mentioned previously that the ganglion cells actually get physically hit by light, but they don't transduce the light. The light is actually caught or absorbed by the photoreceptors, which we mentioned are facing the wrong way. Okay? So the photoreceptors are going to do their cis-trans isomerization for us, and that's going to set up some activity in our horizontal cells. And these horizontal cells that you might see here are going to be monitoring the activity of many different photoreceptors, sometimes rods, sometimes cones, sometimes both. Okay? So we have horizontal cells first, followed by bipolar cells, okay, followed by amacrine cells, and all of that is going to feed into, specifically the amacrine cells in particular, will feed into ganglion cells. Okay? So there's quite a bit of information that we have here, quite a bit of, you might call it neural processing, that goes from the point of transduction to the ganglion cell response. And I would like to point out something very important about the ganglion cells. In fact, a lot of important points about the ganglion cells, but here's one that I'd really like you to capture. These ganglion cells are cells like the ones we've talked about many times before in class. You'll remember that we have dendrites, we have a cell body, and we have axons. The reason that this is so important for ganglion cells is that the axons that we have in ganglion cells are going to be shown right up here, and right up here, and right up here. This whole layer is ganglion cells. And their axons are going to form the optic nerve. And the optic nerve is going to be the nerve that transmits information from the eye back to the thalamus and then eventually all the way back to the occipital lobe. So we have to somehow get information out of the phototransductory area back to the thalamus and back to the brain. And this communication cable, you can almost think of it as a telephone line, will be the axons of these ganglion cells. Okay? So these are very important. And very interestingly, these ganglion cell axons will form this optic nerve, which actually is the second cranial nerve. The cranial nerves are numbered, and we'll get into different kinds of cranial nerves as we go through the course. The second cranial nerve is the optic nerve, and this will exit your eye on the relatively nasal portion of your eye, so the portion closest to your nose. Okay? And that's where we have that optic disc that we talked about earlier. The optic disc is actually the ensemble of axons leaving the eye, and it creates this perceptual phenomena that we call the blind spot. Okay. So ganglion cells are especially involved in extracting what we might call meaningful information. You might recall from a previous video that we had when we talked about information, we had described information as the reduction of uncertainty. The photoreceptors catch the light, but the ganglion cells play an important role in reducing uncertainty about what's out there in the real world. And they do this by providing really important information about boundaries and edges. And we'll see in just a moment how they do that. So they're not catching the light and transducing it for us, but they're helping us to understand where the boundaries are, where the dark portions of our scene are, and where the light portions are. And if we go to move around, and we'll recall that J.J. Gibson reminded us that vision is for action, we need to figure out where to step. And some steps are better than others. We need to figure out where is it light, where is it dark. The ganglion cells are going to help us do that. So typically, objects in the environment differ from each other in their surroundings with respect to the intensity of light. If you look at this scene right here, the bluish area is relatively dark, the white area is relatively light. That boundary information between the dark blue and the light white will allow you to read letters in this particular case. It will allow you to step down the stairs. You can see slight differences in the reflectance of light, and figuring out where those boundaries are is really very important. The ganglion cells specialize in detecting these luminance differences, these luminance edges and so forth, and they're going to report that information to subsequent regions of our central nervous system. Okay, so location, location, location is an important feature that we have here. Photoreceptors face away from the incoming light. They are the most posterior of the three layers that we've talked about. Within the middle layer, the horizontal cells are closest to the photoreceptors, the bipolar cells are next, and the amacrine cells are furthest away from the photoreceptors. The ganglion cells are going to be this very last stop before we have this neuronal signal exiting the eye and making its way off to the thalamus. A very interesting fact, I think, about all of this biological complexity, the different kinds of photoreceptors, the middle layer cells, the ganglion cells, all of that is no thicker than a postage stamp. So here I'm holding a postage stamp in this orientation, but I can hold it just along its width, and that small width would be about the width of your retina. And we have all of these different cell types operating in that very, very small width. In future sessions, we'll be talking about how evolution might have been able to generate that level of biological complexity in such a small space. But I hope you'll just appreciate the tremendous complexity that we have in such a small unit of space. The retina is no thicker than a postage stamp. Okay, so all these cell types that you see here, all of this is happening in something no thicker than a postage stamp. Here's a little memory aid that might help you for an upcoming test that we might have. I think back to my time in New York City, and I used to eat a lot of bagels because I'm a vegan, and bagels tend to be vegan, and there are lots of different bagel shops around. I had a hypothetical bagel shop, which I called RH Bagels. And bagels is spelled this way, but if you do spell it this way, which is a unusual spelling for the bagels that we would normally eat, the RH Bagel Shop will help us recall this particular sequence. And it's a sequence of events that happens from the point of phototransduction to the point at which that signal leaves the eye and heads out toward the thalamus. So we have receptors first, they catch the light, then we have these middle layer cells, and in sequence, functionally, they are horizontal cells, bipolar cells, amacrine cells. Ganglion cells would be the last stop inside of the retina. Then out along the optic nerve, we're going to wind up going to the lateral geniculate nucleus. That is a particular nucleus inside of the thalamus. And then from that thalamus off to the striate cortex, or the primary visual cortex, which is all the way back here in our occipital lobes. Okay? So from the point of phototransduction to activity now all the way back in our occipital lobes, we have this relatively simple mnemonic, RH Bagels. RH Bagels is spelled out here: receptors, horizontal cells, bipolar cells, amacrine cells, ganglion cells, optic nerve, lateral geniculate nucleus inside of the thalamus, and striate cortex inside of the occipital lobe. Okay, ganglion cells, as we've mentioned a few times, don't respond directly to light. They respond to the output of middle layer cells. Okay? And there are approximately 1.25 million ganglion cells per eye. So you can tell that we have many, many more photoreceptors then we have ganglion cells. There's a many-to-one mapping in that relationship. And some people have talked about this as a form of data reduction, and it's approximately a 100-to-1 data reduction within the retina. So lots of photoreceptors, and they're going to be reduced to just a couple of ganglion cells that essentially survey, in many cases, many photoreceptors. By analogy, ganglion cells would summarize a 750 to 1,000 word essay into just 7.5 to 10 words. And in a week or so, you'll have your first writing assignment for this course, and I might ask you to write something like 1,000 words or so. If you can imagine writing that essay, and then having just a 10-word summary of that, that would give you an appreciation for the level of data reduction that we have from the photoreceptor level, where light is being caught, to the ganglion cell level. Okay, electrophysiological recordings are conducted to determine ganglion cell receptive field properties. We've introduced receptive field properties once before in our course when we talked about biological information processing. The receptive field area of the photoreceptor mosaic to which a ganglion cell responds is its receptive field. Okay, so we can think of the receptive field as the region of space to which the cell is responding. And because light travels in straight lines, there's a correspondence between a region of space and a patch on the retina. Okay? So we can try to map out these receptive fields of a ganglion cell. The receptive field size is smallest in the fovea, in the center of the macula, or the center of the retina, and the smaller the receptive field size, the sharper the vision. And we had seen previously the Hermann grid, which gave us this illusion of dots in our periphery, but then when we looked at any one of those dots on the Hermann grid, it went away. Any one of the dots would go away. And this is because the receptive field size is changing. Okay, the ganglion cells are also characterized by a very general neuroscience principle that we call center-surround antagonism. Ganglion cells exhibit center-surround antagonism. Stimuli that excite the center suppress the surround, and vice versa. And in just a moment, we'll show you some of those positive and negative diagrams that we had seen once before in our conversation about biological information processing. That's a type of center-surround antagonism. And we have this going on within ganglion cells. It's a type of lateral inhibition, which we might define this way. Lateral inhibition is some type of antagonistic neural interaction between adjacent regions of a sensory surface such as the retina. And we'll show you an example. Okay? In just a moment, we'll get to the example. You've seen something like this before. For ganglion cells, there are two types of center-surround antagonistic arrangements. We've seen on-center cells and off-center cells. On-center and off-center cells are equally numerous inside of the ganglion cell population, and their axons, interestingly, remain segregated through the next stage in the visual pathway, which is going to be off to the thalamus. And you might recall, within the thalamus, there's a particular relay station that's called the lateral geniculate nucleus, or the LGN. So we have these axons coming out of the ganglion cells, and some are on-center cells, some are off-center cells. And those axons stay relatively segregated, on-center versus off-center, very interestingly. Okay, so here's what I promised. Here's our diagram of the receptive field. This is the on-center ganglion cell. So this is not a photoreceptor. Right? This is a couple of stages past the photoreceptors, but still inside the retina. We still haven't gotten out of a width equivalent to the width of a postage stamp. Okay? And this particular cell will fire many action potentials if the light is distributed in this way. A lot of light in the center, where you see these positive signs. And then we have a darker surround, where we have these negative signs. Okay? So on-center cells respond maximally to light increments in the center and light decrements in the surround. Okay? So an on-center cell might respond maximally to this kind of stimulus. It looks a little bit like a donut. It's light in the center, dark in the surround. Okay? And this cell would absolutely love this kind of a stimulus, and it would actually hate this kind of a stimulus. So you can be recording from this neuron, and here you can get a lot of action potentials. Then just as we're recording from this, we change the visual stimulus from this to that, and the cell stops. We turn it back over to here, and it goes. We turn it over to here, and it stops. This would be the activity of an on-center cell. So what's really, I think, interesting about this is that these cells are signaling how light is distributed out there, and it lets us know where the dark regions are, where the light regions are. And all this, again, is happening inside of the retina, no thicker than a postage stamp. The The other variety just has the reverse configuration. We have minus signs in the center. We have plus signs out here in the surround. We call this an off-center ganglion cell. It responds maximally, and what we mean by that is it fires action potentials maximally to light decrements in the center and light increments in the surround. Okay, so this has now a lot of responding to this. It would love this dark center, light surround, and that's firing away, and we change the stimulus to this, and now the cell stops. We change it back, the cell starts up again. Oops, excuse me. There we are, okay? So it's firing a lot to this, and then it stops in response to that, okay? Light center will turn this off. So we have these two different populations of ganglion cells, which are maybe tuned to the same region of space, but they're reporting whether we have some sort of light increment in the center or instead a light decrement in the center. And so now the ganglion cell population can begin offering different population responses to tell us how the light is distributed. Remember that the photoreceptors don't do that. They either offer us a cis-trans isomerization or they don't. Now we have two different varieties of ganglion cells that can begin to reduce the uncertainty or provide information about how the light is distributed. Ganglion cells don't have any particular orientation preference. So in this diagram, what I'm showing you is that we could put up a dark bar that might be vertically oriented, or it might be horizontally oriented, and this cell might have some minimal response to this one and a minimal response to that one. Maybe not entirely zero, but close to zero. It wouldn't have a differential response to these different orientations. Later in the visual pathway, and in our next lecture, we'll be talking about cortical cells. We see the emergence of cell types that are tuned not just to light centers or dark centers, but to particular orientations of light. The ganglion cells, though, at least in the human, are not orientation selective. Okay? An important point, and this would be one of today's mantras, is that the visual system responds to contrast. Let's all say that together. The visual system responds to contrast. Now, what's a little confusing about that is the visual system also responds to light, but we're going to make a distinction here between light and contrast. Light is simply photons, right? And we have photoreceptors, cone types, different rod types that are going to do the cis-trans isomerization to catch that light. But what we really need to know if we're going to start moving around is where is the contrast? Where is the level of where are the high levels of light? Where are the low levels of light? This is something that's going to be very important to us. And here's an important thought exercise that I read about through a very beautiful writing by J.J. Gibson. J.J. Gibson asks us to think about being in a sauna or a really, really thick fog. And if you're in a sauna and the fog is so thick in there and you put your hand in front of your eyes, you might not even be able to see your own hand. If somebody throws the light switch on or off, you might be able to tell that the room gets lighter or darker. So you can somehow transduce that there's a certain ambient level of light, which is relatively high or relatively low, and yet you still can't see your hand. He has us think about this because he's making the distinction between being sensitive to light and being sensitive to contrast. When I hold my hand in front of my shirt, my hand is slightly lighter than my shirt is. There's some luminance contrast there. So I now get some image information. I can say that my fingers are positioned in a particular way. I'm not just getting light, I'm actually getting some boundary information. Where is it light? Where is it dark? I'm contrasting light and dark areas. The visual system responds to this kind of a contrast, and ganglion cells are particularly good at that. It's important that we can quantify this contrast, and there's a very simple formula for doing it. This is called the Nicholson contrast formula, and it is requiring only some simple addition and subtraction and one division, or one ratio. We introduce some terms called Lmax and Lmin, and Lmax simply corresponds to the luminance level at its maximum and the luminance level L at its minimum. So in this particular diagram, we can see that we have different levels of light. Total numbers of photons coming off of these white letters will typically be greater than the total number of photons coming off of this relatively dark patch that we have here. So we can ask, what is the maximum level of light, Lmax? What is the minimum level of light? And we find the difference of those two quantities, and that becomes the numerator of our Nicholson contrast equation. What we can then do is take exactly those two quantities and now add them. We have the difference over the sum. We'll take that ratio, and this is a way of expressing how much contrast we have out here in the distal stimulus that is out here in the actual environment. Okay? So let's take a particular example. Imagine that we had L max equaling 99. And imagine that we had L min equaling 1. That is to say, hypothetically, we had a light level of 99 coming off of the white regions, and we have a light level of 1 unit coming off of these dark regions. We would do 99 minus 1. That would give us 98 in the numerator. We'll take those numbers down here. 99 plus 1, that'll give us now 100. So we have 98 over 100. We would then have a Nicholson contrast of 98%. So that would be a relatively high contrast level, 98%. If there were absolutely no contrast, that is to say, if the light were completely homogeneous, that there was no differences in the light levels across our visual world, we'd be at 0 contrast, and we could still have cis-trans isomerizations, but our ganglion cells would shut up because there would be no on-center, off-surround distinction to be made in that level. So we do need to have some kind of contrast, and the Nicholson contrast formula is a mechanism by which we can quantify the physical stimulation out here. Okay? Here's an example of some high contrast stimuli. I have a really dark in the center and a really light in the surround, and conversely over on that side. If I now take the contrast away a little bit, you might notice that I still have a dark region and a light region, but this is now not quite so dark, a dark gray, and this is not quite so light, a light gray. And there's the reverse configuration. And then I can go all the way to here, where inside of this gray region, you might imagine that there's a center area, but you can't see it because there's no contrast. Over here I have a slightly darker circle, and there might be a center region, again, that could be slightly different from its surround, but here I have 0 contrast. So no difference between the center and surround, unlike this and this. Okay? All right, so that was our introduction to ganglion cells. Ganglion cells help us extract these boundaries, and they exist inside of the retina a few stages past the point of phototransduction. After the ganglion cells have made their response to the information that they're receiving from the middle layer cells, they send that information across the optic nerve out to the thalamus, specifically the lateral geniculate nucleus, and then off to the cortex. What I'd like you to do is make some notes about what you thought was particularly clear in this section, and also what was particularly unclear. And you can bring those notes to class, and we'll have some conversation about that. See you in class.